Hello and welcome everyone. Myself Mizanur Rahman, and today my topic of discussion is a red blood cell or RBC. Today we will discuss about introduction, normal value, morphology, properties, lifespan, fate, functions, variation in number, variation in size, variation in shape, and variation in structure. So let us introduce. RBCs are the non-nucleated formed elements in the blood. RBC are also known as erythrocytes. So RBC are one of the cells which are generally non-nucleated, and the red color of the RBC is due to the presence of the coloring pigment called hemoglobin. And RBC also plays a very important or vital role in the transportation of respiratory gases like oxygen, carbon dioxide, etc. RBC are in larger in number as compared to other blood cells, likely WBC and platelet. What are the normal value of RBC? RBC count generally ranges from 4 to 5.5 million per cubic mm of blood. In case of male, it is about 5 to 5.5 million, and in case of female, it is 4.5 to 5 million per cubic mm of blood. Now, morphology of red blood cells. What is the normal shape of the RBC? The RBC is dumbbell shaped or biconcave shaped. The central portion is generally thinner and the peripheral portion is thicker. The biconcave shape of RBC has some mechanical and functional advantages. What are those advantages? This biconcave shape helps in equal and rapid diffusion of the oxygen and other substances into the interior of the cell, and it also provides the larger surface area which helps in more and more of absorption and removal of different substances. The minimal tension is offered on the membrane when the volume of other cell alter, and because of this biconcave shape, while passing through the minute capillaries, RBC squeeze and through the capillaries, they pass very easily without getting any damage. So one is first opportunity or advantage is rapid diffusion of gases. Second is large surface area. Number three is minimum tension, and number four is passing through the minute or little capillaries. Now normal size. The diameter of RBC is 7.2 micrometer, but it may ranges between 6.9 to 7.5 micron also. Thickness. At the periphery, the thickness is 2.2 micron, and in the center it is 1 micron. The difference is due to the biconcave shape. Surface area 120 square micron and volume is 85 to 9 cubic micron. Now normal structure. RBC are generally non-nucleated, but there is one exception. In case of camel, they got the nucleated RBC. You can see the diagram of RBC. Diagram A is surface view of the RBC, and diagram B is a section view of RBC. But in case of human being, they don't have any nucleated RBC. So the DNA is also absent, and other organelles such as mitochondria, Golgi apparatus are also absent in RBC. Because of the absence of mitochondria, the energy which is needed for them to maintain their cellular lifestyle, these are produced in glycolytic process, and RBC don't have any insulin receptor, and so the glucose uptake by this cell is not controlled by insulin. So the glucose uptake has no relation with the insulin. RBC has also a special type of cytoskeleton which is made up of actin and spectrin. Both these proteins are anchored to transport proteins by means of another protein called ankyrin. And these ankyrin, spectrin, actin, these type of protein also help to maintain that biconcave shape also. Absence of spectrin result in hereditary spherocytosis. In this condition, what happens? The cell is deformed, loses its biconcave shape and becomes globular or spherocytic. The spherocyte is very fragile and easily ruptured or hemolyzed in hypotonic solution. Now, properties of red blood cell. Rolex formation. When blood is taken out of the blood vessel, the RBCs are piled up one above another like the pile of coins. This property of RBC is called Rolex or plural Rolleau formation. It is accelerated by plasma proteins, globulin and fibrin. This is Rolex formation. That diagram you can see figure 9.2. Now specific gravity, it is 1.092 to 1.101, and packed cell volume. Packed cell volume, what is this? It is the proportion or percentage of blood occupied by RBC in percentage. So why in the percentage form we can calculate the in which percentage the RBC is present. Generally it is 45% in the blood, and it is also known as hematocrit value. In case of male it is 45%, and in case of female it is little bit lesser, 41 to 42%. So next, suspension stability. During circulation the RBC remain in suspended uniformly in the blood. The property of the RBC is called suspension stability. Now, lifespan of red blood cell. Average lifespan of red blood cell is about 120 days. After the lifetime, the old or senile RBCs are destroyed in reticuloendothelial system. Now, determination of lifespan of RBCs. How we can basically determine the lifespan of RBC. For the determination of lifespan of the RBC, we use radioisotope method, where the RBCs are tagged with radioactive substances, and like the radioactive iron or radioactive chromium, and life of RBC is determined by studying the rate of loss of radioactive cells from circulation. Okay. Now, fate of red blood cell. We will discuss little bit about this portion. We will discuss further in more videos or more details in another video. The fate of RBC is very important topic, so we will discuss this in next and another video very deeply. But we will generally discuss this here. When the RBC become older or become the 120 days, the cell membrane become more fragile, rigid. Then diameter of the capillaries is less or equal to that of RBC. Now younger RBCs can pass through the capillary very easily as they are more expandable or not that much fragile. That's why they can pass very easily. But as these are in fragile nature, they can't pass through these narrow membranes. The older cells are destroyed while trying to squeeze through the capillaries. The destruction occurs mainly in the capillaries of red pulp of spleen. Because of the diameter of the splenic capillaries is very small, so the spleen is called as the graveyard of RBCs. Destroyed RBCs are fragmented and hemoglobin is released from the fragmented parts, and hemoglobin is immediately phagocytized by macrophages of the body, particularly the macrophages present in the liver or Kupffer cell and spleen and bone marrow. Now, hemoglobin is degraded into iron, globin and porphyrin. Now, iron combines with the protein called apoferritin to form ferritin, which is stored in the body and reused later. Globin enters into the protein depot for later use, and porphyrin is degraded into bilirubin, which is excreted by liver through bile. Daily 10% of the RBCs which are senile or old are destroyed in normal young healthy adults. It causes release of about 0.6 gram per dl of hemoglobin into the plasma. From this 0.9 to 1.5 milligram per dl bilirubin is formed daily. So this is normal range, but above this is a case of a disease condition. If we take a snapshot, then destruction of RBC in spleen, how it has happened? Then release of hemoglobin occurs. From this part, there are three fragments appears. One is iron part, one is globin, and one is porphyrin ring. Now, when iron is attached with apoferritin protein, it is converted into ferritin and stored and reused later. Globin is also stored in protein pool and stored and reused, but porphyrin is converted into bilirubin, which is excreted through the excreta or stool. Now, functions of RBC. What are the functions that are generally performed by red blood cell or erythrocytes? So, first of all, most important function, transport of oxygen from the lungs to the tissue. First of all, what happens? Our body need oxygen to survive, because without oxygen we can't survive anymore. Oxygen is the basic necessity. So what hemoglobin does? Hemoglobin combines with oxygen to form oxyhemoglobin, and about 97% of the oxygen is transported by blood in the form of oxyhemoglobin. This is also a specific pathway we will discuss in the another video. Now, transport of carbon dioxide from the tissue to the lungs. As all of us we know, carbon dioxide are produced in the inner part of our body or in the tissue cells, and these are must to be removed. If we does not remove these particles, then it can create toxicity in our body. Hemoglobin combines with carbon dioxide to form carb hemoglobin, and about 30% of the carbon dioxide is transported in this form. Now, and RBC also contain a large amount of Ca or carbonic anhydrase. This enzyme is necessary for the formation of bicarbonate from water and carbon dioxide. Thus, it helps in transport of carbon dioxide in the form of bicarbonate from tissue to lungs, and about 63% carbon dioxide is transported in forms. So, RBC helps in two parts. RBC directly carries some carbon dioxide, and RBC helps to convert some carbon dioxide into bicarbonate ion and convert through the RBC blood flow or bloodstream through the capillaries. And in this process, carbonic anhydrase enzyme helps there. Now, buffering action in blood. The hemoglobin functions as a good buffer. By this action, it regulates generally the hydrogen ion concentration and thereby plays a major role in the maintenance of acid-base balance. And in blood group determination, RBC carry the blood group antigen like antigen A, antigen B, and Rh factor. This helps in determination of blood group. Also, we will discuss in another next any more videos how blood groups are determined and what are the blood groups, A positive, B positive, AB positive, O positive, O negative, AB negative. This type of blood groups we also discuss in another video. Now, variation in number of red blood cells. So, first of all, physiological variation. There are some physiological variation like increase in RBC count. Increase in RBC count is known as polycythemia. So, what is this term called? Polycythemia is what? Polycythemia is increase in the RBC count is known as polycythemia. It occurs in both physiological and pathological conditions. When it occurs in physiological condition, it is called physiological polycythemia, and when it is when it is caused by pathology, then it is known as pathological polycythemia. The physiological polycythemia is temporary or marginal condition. It occurs in the following condition like age. At the time of birth, the RBC count is 8 to 10 million per cubic mm of blood. The count decreases within 10 days after birth due to destruction of RBC causing physiological jaundice in some newborn babies. However, infants and growing children, the cell count is more than the value in adults. 6. Before puberty and after menopause, the RBC count is similar to that of males, but during the reproductive period of female, the count is less. That is nearly about 4.5 million per cubic mm. High altitude. Inhabitants of the mountains have an increased amount of RBC generally more than 7 million per cubic mm, it is due to the hypoxia condition. What is hypoxia? It is decreased oxygen supply to the tissue in high altitude. Hypoxia stimulates the kidney to secrete a hormone called erythropoietin, which will further stimulate the red bone marrow to secrete to RBC. Basically, it enhances the enhances the process of erythropoiesis, which will further give birth to the red blood cell or erythrocytes. Now, muscular exercises. There is some temporary increase in RBC count after the exercise. It is because the mild hypoxia and contraction of spleen. Spleen stores RBC and hypoxia increases the sympathetic activity, resulting in the secretion of adrenaline from the adrenal medulla, and adrenal contracts spleen and RBC released into the blood. Now, emotional condition. When a person is generally in anxiety or depressed, then through the help of sympathetic activity, the RBC count is increased. Increased environmental temperature. When the atmospheric temperature is increased, then RBC is also increased. As there is increased temperature, increase all the activities of the body, including production of RBCs also. After meals, there is also a slight increase in the RBC count after taking meals. It is because of need of more oxygen for metabolic activities. Now, if we take a flow chart, high altitude, which will create hypoxia condition, which stimulate the kidney and which will help to production of erythropoietin hormone, which stimulate the bone marrow and which will help in production of RBCs. So this is physiological polycythemia in high altitude. Now, emotional condition and muscular exercises increases sympathetic activity, which will further increase secretion of adrenaline, which will contract the spleen and release RBC. This is also a physiological polycythemia in emotional condition and exercises. Now, decrease in RBC count. When now we have known about the increase in RBC count, now we will go to know the decrease in RBC count. When the decrease occurs, first of all, high barometric pressure. When we go in high barometric pressure in deep sea, when oxygen tension of blood is higher, then RBC count is decreased. During sleep also the RBC count is decreased because the all body activity is decreased during sleeping time. Pregnancy. In the time of pregnancy, RBC count is also decreased because it increases in ECF volume, and in the ECF volume it increases the plasma volume, also resulting in the hemodilution. So there is relative reduction in the RBC count. In pregnancy, red blood cell count is not decreased, but the plasma is increased. That's why relatively it seems that the RBC is decreased. Now, pathological variations. Pathological polycythemia. Pathological polycythemia is the abnormal increase in the RBC count. Red blood cell count may be increased above 7 million per cubic mm of blood. Polycythemia is of two types. There are primary polycythemia and secondary polycythemia. What is primary polycythemia? Primary polycythemia is known as polycythemia vera. It is a disease characterized by persistent increase in RBC count over 14 million per cubic mm of the blood. This is always associated with increased white blood cell count above 24,000 per cubic mm of blood. Polycythemia vera occurs in myeloproliferative disorder like malignancy of red bone marrow or cancer of red bone marrow. If the tumor occurs in the bone or the cancer occurs in the bone, then these types of disease may occur, and this will increase in huge amount of production of red blood cell, white blood cell, etc. Now, secondary polycythemia. This is secondary to some of the pathological condition disease such as respiratory disorder like emphysema, congenital heart diseases, Erasure disease like condition which associated with hypertrophy of right ventricle and obstruction of red blood flow to lungs. chronic carbon monoxide poisoning, poisoning by chemicals like phosphorus and arsenic, repeated mild hemorrhages. All these type of conditions lead to hypoxia, which stimulate the release of erythropoietin. Erythropoietin stimulate the bone marrow, resulting in increased RBC count. Now anemia. Anemia is what? It is abnormal decrease in RBC count is called anemia. We will discuss more about in the another video, because these are not a short topic, these are high yielding topics, so we will discuss in another part of the video. Now variation in size of red blood cell. Under the physiological conditions, the size of the RBCs in venous blood is slightly larger than those of arterial blood. In pathological condition, the variation size of RBCs are microcytes, macrocyte, anisocyte. Microcyte means smaller cell, macrocyte means larger cell, anisocyte means cell with different sizes. Microcyte, in which type of condition microcytes or red blood cell become smaller? Microcytes are present in iron deficiency anemia, prolonged forced breathing, increased osmotic pressure in blood, and macrocyte occurs in megaloblastic anemia, decreased osmotic pressure in blood, and anisocyte occurs in pernicious anemia. These type of conditions will explained in anemia video. Variation of shape of red blood cells. Now, shape of the RBCs is altered in many conditions, including different types of anemia. Crenation, shrinkage as in hypertonic condition. Spherocytosis, globular form as in hypotonic condition. Elliptocytosis, elliptical shape in certain types of anemia. Sickle cell, crescentic shape as in sickle cell anemia. Poikilocytosis, unusual shapes due to deformed cell membrane. The shape will be a flask, hammer, or any unusual shape. Variation in structure of RBC. Punctate basophilism, striated appearance of RBCs by the presence of dots of basophilic materials like porphyrin is called punctate basophilism. It occurs in conditions like lead poisoning. Ring in red blood cell, ring or twisted strands of basophilic material appears in the periphery of the RBCs. This is also called the goblet ring. This appears in RBC in certain types of anemia. Whale jolly bodies. In certain types of anemia, some nuclear fragments are present in the ectoplasm of the RBCs. These nuclear fragments called whale jolly bodies. So this is for now. We have covered a major topic today. We have discussed about the RBC part or red blood cell, general discussion of red blood cell. Further discussion will occur in another videos. I think I have covered all the concept very clearly. If you have any doubt or any misconception, please ask in the comment section. I will try to reply them. And do like, share and subscribe. And thank you for watching.